Assalamu alaikum to all of you. I am Umar Qureshi. I am the team member of the Medimartt. Medimartt is a platform for the medical students. As you all know that we are regularly trying to share some short videos on different topics. So here we come with the another video. This is a short video on the blood part. Okay, this is the fourth part of the blood. Okay, so please watch our previous three parts, then only you will understand this part. Okay, so please check it out, and please guys, please watch this video. Okay, and don't skip in between, because if you will skip you will not understand anything. So please watch it full. Okay, and please try to share it as much as possible. Please guys, subscribe to our channel, please like this video, and please comment down anything you need like in the comment box. Okay, so let's start. So basically this is the slide. Okay, and in our previous videos we have discussed till the sites of erythropoiesis. So today we will discuss about the stages of the erythropoiesis. So there are mainly 6 stages of erythropoiesis, and those are the proerythroblast, the basophilic erythroblast, the polychromatic erythroblast, the orthochromatic erythroblast, reticulocyte, and the last one is the erythrocyte. Okay, so we will start with the first one, that is the proerythroblast. So in proerythroblast the cell size is 15 to 20 micron in diameter. Okay, the nucleus, it is crescent and the pale staining, and it is large and round. Okay, the nucleoli, it is chromatin, form a fine stippled reticulum. Cytoplasm, it is deep violet blue, that is basophilic, and hemoglobin is absent over here, and mitosis occurs over here. Okay, so you can see this is the proerythroblast. This is a handmade diagram by our teammate. Okay, so it is the proerythroblast. This blue color is fully like a cytoplasm. Okay, that is why it is basophilic, and hemoglobin is absent over here. Mitosis is like occurring over here. Okay, that's the thing. Now moving on towards the next thing, that is the basophilic erythroblast. Basophilic erythroblast, the cell size of it is the 12 to 16 micron in diameter. The nucleus, here condensation occurs, the nucleoli is disappeared. Okay, and cytoplasm, it is deep violet blue, again the basophilic, and hemoglobin, it begins to appear. Okay, and mitosis is active. Okay, just like that. This is the basophilic erythroblast, you can check it out. Okay, now moving on towards the third stage, that is the polychromatic erythroblast. Now what is polychromatic erythroblast? Okay, so basically polychromatic, poly means many and chromatic means colors. Okay, so many colors, so that's why it is called as the polychromatic erythroblast. So first of all, the cell size of it is the 10 to 14 micron in diameter. Nucleus, it is more condensed. Okay, let's see what that means. So nucleus, this is the nucleus. Okay, in between it is like more condensed. Okay, then cytoplasm, it is polychromatic appearance. I already told you that poly means different colors. Okay, poly means many and chromatic means color, so you can check it out. This third circle, in it the cytoplasm is a polychromatic. Okay, polychromatic in appearance, and hemoglobin is increased in it, because in our previous stage hemoglobin begins to appear. So that's why in this stage hemoglobin is like increasing slowly. So in this hemoglobin increased, and mitosis is again active, just like that in our previous stage. Okay, so moving on towards the next stage, that is the stage 4, orthochromatic erythroblast. Okay, so cell size in it is the 7 to 10 micron in diameter. Okay, as we are moving towards the more stages. Okay, so according to that, cell size is rapidly decreasing. Okay, so in the first stage it was around 15 to 20 micron in diameter, then in stage 2 it was around 12 to 16 micron in diameter, then stage 3 it was around like 10 to 14 micron in diameter, and like in stage 4 it again decreases, that is about 7 to 10 micron in diameter. Okay, so it was about this stage 4, cell size 7 to 10 micron in diameter. Now let's talk about the nucleus of it. Nucleus of this orthochromatic erythroblast, it is the cartwheel appearance, that is pycnotic ink spot. Okay, so let's see how it looks like. So this is it looks like, it is this nucleus, it is pycnotic. Okay, and cartwheel appearance means it will look like the wheel of a cart. Okay, now the cytoplasm, it is eosinophilic. Okay, It is not basophilic. Basophilic means the blue colored, and eosinophilic means the like a little bit of reddish type of color. That's the thing. Now hemoglobin, it is increased. Why this is becoming the red color? Because hemoglobin is rapidly increasing. Okay, and in this stage the mitosis is stopped. Okay, so you have to understand this thing. Like mitosis was active till the third stage. Okay, and in this fourth stage mitosis is stopped. Okay, so your teacher may ask you like at which stage the mitosis is like terminated. So you have to say it is the orthochromatic erythroblast. Now let's talk about pycnosis. What is pycnosis? Pycnosis is basically degeneration of nucleus which breaks up and finally disappear due to the lysis. Due to the lysis, this nucleus disappears. Okay, and degeneration of it occurs. So that is called as the pycnosis. Now moving on towards the stage 5, and that is the reticulocyte, that is the young red cell. Okay, so this is the young red cell. Okay, after it the red cell becomes the mature. Okay, so that's why this stage is called as the young red cell, or it is also called as the reticulocyte. The cell size of it is the 7 to 8 micron in diameter. Nucleus, in this nucleus disappeared because as we saw in the previous stage pycnosis occurs, and pycnosis was the degeneration of the nucleus. So that's why in the stage 5 nucleus will completely disappeared. Okay, and cytoplasm, here it is eosinophilic, and hemoglobin, as we are moving towards like more stages. Okay, so the hemoglobin is increasing rapidly. So in this the hemoglobin is more increased, and mitosis is stopped as it was like in the previous stage. Okay, moving on towards the stage 6th, that is the erythrocyte, or it is also called as the mature red blood cell. The time requirement for this stage is like pro-normoblast to reticulocyte is 7 days, and from reticulocyte to erythrocyte it is around 2 days. So what is like pro-normoblast? Okay, so pro-erythroblast or the pro-normoblast is one and the same thing. So from the pro-normoblast, that is the stage 1, to the stage 5, that is the reticulocyte. Okay, it took around the 7 days to form from the pro-normoblast to reticulocyte, and then from reticulocyte to erythrocyte it took around the 2 days. Okay, so that's the thing about the stages of the erythropoiesis. Okay, and let's move, let's watch this figure. It is the figure of Guyton. Okay, genesis of RBC, red blood cell. So the first stage was the pro-erythroblast, the second was the basophilic erythroblast, the third one is the polychromatophil erythroblast, the fourth one orthochromatic erythroblast, and the fifth one is the reticulocyte, and the last one, that is the mature RBC, which is also called as the erythrocytes. You can see it in the figure. Okay, that's the thing. Now moving on towards the basic changes or major changes in the erythropoiesis. So the basic changes or major changes in the erythropoiesis are, so a gradual reduction of cell size and alteration of shape. Okay, maybe like cell size may reduced. Okay, and alteration of shape can occur also. Okay, so that's the major change or the basic change. Now maybe disappearance of nuclei and nucleus also can occur over here. Okay, actually not maybe, these are the things that will occur in the erythropoiesis, in the formation of the mature red blood cell, like from pro-normoblast to the erythrocyte. These all things will occur, as we saw that in the pro-normoblast or pro-erythroblast the cell size was the 15 to 20 micron in diameter. So it was gradually reducing. So that's the point, a gradual reduction of cell size and alteration of shape will occur. Okay, then disappearance of nuclei and nucleus, we saw that the degeneration of the nucleus occurs. Okay, so that's why disappearance of the nuclei and nucleus. Then the appearance of the hemoglobin and cessation of cell division. Cell division is also called as the mitosis. Okay, so as we saw in the third stage mitosis was stopped. Okay, so that's the thing here, the appearance of, and like as we saw the hemoglobin was rapidly increasing, so that's called as the appearance of hemoglobin. Okay, then change in the staining reaction of the cytoplasm from basophilic via polychromatophilic to eosinophil. As we saw in the first stage the cytoplasm was basophil. basophilic. Okay, then in the second or third stage, basically in the polychromatophil stage, okay, it was like a polychromatophil, or the cytoplasm was polychromatophil. Okay, then in the last stage, that is the eosinophil, okay, it was eosinophilic. Okay, the cytoplasm was eosinophilic basically. Now moving on towards the next topic, that is the factors controlling or required for the erythropoiesis. Okay, and the factors are the general factors, then the hemoglobinization factors, and the maturation factors. Okay, then there are some more factors, but we will see like it in the later words. Okay, first of all we will see what are the general factors. So general factors involves the hypoxia, erythropoietin, and endocrine hormones. Okay, so what is hypoxia? Hypoxia is basically a decrease in the oxygen level. So it is a potent stimulus for red blood cell production in the bone marrow. It enhances liberation of hormone erythropoietin. Conditions producing hypoxia are the high altitude, when basically we are standing on the high altitude like the mountains and all, we feel the decrease in oxygen. Okay, so that's called as the hypoxia. Now the congenital heart disease. Okay, so congenital heart disease also we feel the hypoxia. Then after hemorrhage also, chronic lung disease, and some anemias also. So that was about the hypoxia. Now moving on towards the erythropoietin. Erythropoietin, in response to hypoxia, kidney releases erythropoietin. When you cause the when you feel the decrease in the oxygen level, so basically your kidney detects it, and your kidney releases the erythropoietin that acts on the bone marrow and stimulate the erythropoiesis. Okay, and this erythropoietin, it acts on the bone marrow, and it in the bone marrow basically it stimulates the erythropoiesis, means the formation of the mature red blood cell. Now endocrine hormones also, these are the general factors like the adrenocorticotropic hormone and the thyroid hormone. So these are the some general factors of for erythropoiesis. Now moving on towards the hemoglobinization factors, and the hemoglobinization factors are the first class protein and the amino acid, like synthesis of the globin portion of the hemoglobin. Okay, so hemoglobinization factors basically it involves the first class protein and amino acids. Okay, then it involves the iron also. Okay, then it involves the copper, cobalt, nickel, calcium, bile salt, vitamin B2, B3, B6, and C. Okay, vitamin C. So first class protein and amino acid, it helps in the synthesis of the globin portion of the hemoglobin, as the in the hemoglobin there are two portions like the heme and the globin portion. Okay, so this first class protein and the amino acid, it helps in the synthesis of the globin portion of the hemoglobin, and the iron, it helps in the synthesis of the heme portion of the hemoglobin. So this heme and the globin together forms the hemoglobin. Okay, now copper, cobalt, nickel, calcium, bile salt, vitamin B2, B3, B6, and vitamin C also helps in the hemoglobinization factors, and these are required for the erythropoiesis. Okay, now there are some maturation factors which help in the DNA synthesis, and those are the vitamin B12, that is also called as the cyanocobalamin, means the other name of the vitamin B12 is the cyanocobalamin, and the other maturation factor is the folic acid. Okay, so this was about the maturation factors. Now there are some more maturation factors, or you can understand it by like writing a note. So vitamin B12, it acts as a coenzyme, and folic acid, it acts as a catalyst in the DNA synthesis. Okay, so in DNA synthesis, vitamin B12, this acts as a coenzyme. Okay, and folic acid basically it acts as a catalyst. Okay, now deficiency of these, means the vitamin B12 and the folic acid, causes the failure of nuclear maturation and cell division. Okay, so basically if all these like vitamin B12 and the folic acid if decreases in our body, then the failure of nuclear maturation and the cell division will occur. So blood contains abnormal, irregular, large, fragile, and oval red cells. This condition is the megaloblastic anemia. When these things like vitamin B12 and the folic acid decreases in our body, so ultimately like a failure of natural like nuclear maturation will occur, and the cell division will not occur in the body. Okay, in that particular thing. Okay, so when all these things occur, simultaneously. So the blood basically contains the abnormal, irregular, large, fragile and oval red cells. Okay, when cell division will not occur, the cell size will not changes. Okay, so according to that, it will be the cell size or the cell will be abnormal, it will be irregular in shape, it will be large, and it will be fragile, and it will be oval. Okay, so what this condition is called? So this condition is called as the megaloblastic anemia. Megalo means the huge or the big. Okay, so that's called as the megaloblastic anemia. Now let's talk about some intrinsic factor of Kessel. Okay, so there are some intrinsic factor of Kessel also. It is produced in the gastric mucosa. This intrinsic factor of the Kessel, it is produced in the gastric mucosa, and these are essential for the absorption of vitamin B12 from intestine to the blood. Okay, so when we eat some kind of food, so this vitamin B12 or like it is absorbed into the blood from this intestine. Okay, so by which thing this vitamin B12 is absorbed into the blood? So the essential factor of it is the intrinsic factor of the Kessel. Okay, it helps in the absorption of the vitamin B12 from the intestine to the blood. Now deficiency of the intrinsic factor also causes, means if like intrinsic factor of Kessel decreases in our body, so this deficiency causes the pernicious anemia, since the vitamin B12 is not absorbed. So ultimately pernicious anemia will cause, because vitamin B12 is not absorbing into the blood. Now the factors necessary for hemoglobin formation. So basically there are some certain factors which are necessary for the hemoglobin formation. Okay, and those are the first class protein and amino acid. I already told you in the previous slide. Now there are some more things. So this mechanism we will understand, like the kidney. Okay, when you feel the decrease in the oxygen level, like which is also called as the hypoxia, so it basically hints the kidney. Okay, so kidney basically secretes the erythropoietin factor, okay, which is also called as the renal erythropoietic factor. Now this renal erythropoietic factor, it basically helps, or it basically work as a catalyst to change the alpha globulin into the erythropoietin. Okay, so this alpha globulin is changes into the erythropoietin with the help of this renal erythropoietic factor. Now this erythropoietin is it basically works on the stem cells, and then these stem cells converted into the proerythroblast, and this proerythroblast it increases the RBC, that is the red blood cells, and this increase in the red blood cells, it increases the hemoglobin concentration, and this hemoglobin concentration basically moves to the tissue organ. Okay, so this is a whole chain, and this tissue organ is related with the kidney also. Okay, so this is a whole chain. This thing today I explained this thing in very short in this video. We will explain this thing more in the next video, inshallah. Okay, so please guys, please watch our more videos, and please subscribe to our channel, because of then only you will get notification when we will upload more videos like this. And thanks a lot.